Welcome back to my channel and welcome to Molecular Biology. In this video, I am going to discuss a very important topic called topology. In the first part, I have discussed what is topology of the genetic material. In the second part, I have pointed out the problems associated with topology and how it solved by topoisomerase. And in the last part, I have discussed how we can distinguish different structures of DNA experimentally. So let's start. Topology is a mathematical term that tells us how we can deform an object smoothly without tearing it. Here in this example, I have deformed a circular structure in different ways like stretching, twisting, bending, and knotting. All these structures are similar topologically, but they are geometrically different. For genetic materials, three parameters are used to describe the topological state. They are linking number, twist, and writhe. Twist for linear objects means bending like this. For helical objects like DNA, one complete turn is called the twist. It can be positive for right-handed helix like BDNA and it can be negative for left-handed helix like ZDNA. The second parameter is called writhe. It defines the number of times double helix cross itself that leads to supercoiling. Suppose this is the double-stranded DNA and when it crosses itself, a writhe is introduced. Writhe can be positive or negative depending on positively supercoiled and negatively supercoiled structure respectively. The third parameter is called linking number and it is the total number of linking within a DNA, or it can be expressed as linking number is equal to twist plus writhe. The value of linking number is fixed until or unless you break open the sugar phosphate backbone. This is the another way by which you can calculate the linking number of a DNA. Now I have solved some numerical problems appeared in competitive examination. You can pause the video to watch how I solved these problems. The first one is straightforward and here the number of turns is given. So linking number is calculated directly. In the second problem, the size of the BDNA is provided and we know that approximately 10 bases are there in one complete turn. So linking number is calculated and it is 10. In the third example, 3 turns are removed from a circular relaxed DNA to make it supercoil. We have to find out the linking number. I have used linking number is equal to twist plus writhe to solve this problem. And in the next part, I will discuss what actually happens during the fundamental process. The two strands are separated in order to initiate replication, transcription, recombination, and cell division. When strands are separated, topological strain is introduced that makes more coil structure ahead. And if this continues for a longer duration, the process will stop. To solve this problem, cell use topoisomerase or in short called topo. They break open the sugar phosphate backbone and rotate it and then resealed it to remove the topological strain. There are generally two types called topo 1 and topo 2. You can find out the difference between topo 1 and topo 2 in the table. So, if we can block the function of topoisomerase, cell will die due to DNA supercoiling. That's why several drugs and antibiotics that target topoisomerase are used to treat cancer and microbial infection. And finally, what will happen if we can increase the concentration of ETBR or other intercalating agents in a tube where covalently closed circular DNA or plasmid is present? Sedimentation velocity as well as writhe will change as described in the picture. Writhe is negative at low concentration and then it increases as the concentration is increased. And if we run relaxed plasmid and supercoiled plasmid in gel, supercoiled structure will run faster than the relaxed plasmid because the structure is more compact than the relaxed one. Plasmid migrate more distance when the linking number is decreased. So that's all for this season. If you like this video, don't forget to share, you can comment and don't forget to subscribe my channel for more update and thanks for watching.